Hello, I'm Chris Bass. I'm a Professor of Applied Entomology at the University of Exeter. Entomology is the study of insects, and I use my time trying to understand how insects become resistant to the chemicals that we use to try to control them.
Hello, I'm John Vondas. I'm professor at the Agricultural University of Athens and researcher at the Institute of Molecular Biology and Biotechnology in Crete. I try to understand the mechanisms by which insects, agricultural pests, and mosquitoes develop resistance to insecticides, aiming to develop new means of managing and overcoming this resistance.
In this video, we're going to tell you about how we detect insecticide resistance in insect pest populations using diagnostics. The emergence of insect populations with resistance to insecticides is a really important and growing problem. It makes insect populations much harder to control, and it reduces the number of insecticides that we have available to use against them. If we are to understand which pest populations are resistant and what they're resistant to, and in turn make decisions about which insecticides we can use for control, we need to be able to detect and diagnose resistance rapidly. One way we can combat the emergence of insecticide resistance in pest populations is to use resistance monitoring programs. These aim to establish the sensitivity of pest populations to insecticides used for control. However, to do this, we need to be able to detect resistance in the field. So how can we detect and monitor insecticide resistance? There are two main ways to detect insecticide resistance in pest populations. The first uses an approach called insecticide bioassays. In this method, insecticides are applied to the pest insects, and their response to that exposure is measured. So how many insects remain alive and how many die.
The procedure for bioassay monitoring of insecticide resistance includes the preparation of insecticide solutions and application on treated surfaces or incorporations in the insect diet using proper safety precautions, absolutely essential when dealing with insecticides. The capture and placement of insects or mites in small glass vials or petri dishes where they are exposed to the insecticides by different means, for example, by contact with the spray surface, by digesting insecticide in food or water solution, or by receiving a specific insecticide dosage in a small droplet in the thorax via a syringe. Then insects are transferred in a clean cage with food and water applied. And finally, the response to the insecticide is measured typically as mortality 24 or 48 hours after the insecticide application. The outcome of the bioassay can be either lethality at a standard diagnostic for resistance dose or quantitative determination of insecticide intensity, usually compared to a known susceptible population. Bioassays are simple, practical, and widely used. They don't require sophisticated equipment and expensive reagents, and the results are often directly relevant to the insecticide resistance phenotype in the field. The feasibility of bioassays depends on whether insects can be easily collected, stored, and grown in the lab, and equally, whether the host plant is easily cultivated in the lab or artificial diets for pests are available. Bioassays have low sensitivity for the early detection of resistance, and they do not detect the underlying resistance mechanisms, which may limit the value of monitoring information, as for example, the cross resistance and the fitness of the trait cannot be assessed.
The other main way we can use to detect resistance is to use molecular diagnostics. These detect resistance markers, the genes or mutations that cause resistance. These can include the identification of an enzyme that breaks down the insecticide or a mutation in an insecticide target which reduces the binding of the insecticide. So how do molecular diagnostics work? First, the insect species is collected from the field. Then, DNA is extracted and a region of that DNA containing the marker is amplified using something called a thermocycler. Following that, the region of DNA is visualized by running it on something called an agarose gel. That allows you to detect if the resistance mutation or gene is present in that individual or not. Molecular diagnostics don't always require expensive equipment and a laboratory. In this example, a test was developed to detect resistance in whiteflies that uses this simple, rapid diagnostic test, and this can be used by the farmer or grower themselves to work out whether the pest population is resistant or sensitive to insecticides.
More specifically, this technology has been developed to measure the expression levels of a detoxification enzyme responsible for neonicotinoid resistance in whiteflies. The procedure for this antibody-based lateral flow test assay includes collection of approximately 10 insects in a tube, addition of a few drops of a buffer solution, and homogenization using a pestle. Addition of the antibodies and the reaction reagents using a droplet, short incubation time for a few minutes, and finally, reading of the visible test results on the strip by eye, where a control line indicates the correct technical procedure and a distinct test line which, if present, indicates that the levels of the detoxification enzymes are high enough to provide resistance.
So what are the value of molecular diagnostics for insecticide resistance management? So molecular diagnostics can provide a lot of useful information. They can be used to determine the frequency of resistance and so understand just how resistant the pest population is likely to be. They can be used to determine its geographical distribution, so where is resistance present and where is it not. Finally, it can be used to understand the cross-resistance spectrum of that pest population, so whether the pest population will be resistant to other insecticides that might be used for control. So we have explained that there are two main ways to detect resistance, but which is the best, bioassays or molecular diagnostics? So the two methods have advantages and disadvantages. In the case of bioassays, these can be used even when the underlying mechanism of resistance is unknown. However, they're often time-consuming to perform and require live material and may not detect resistance when it occurs at low frequency in the population. In the case of molecular diagnostics, these are highly sensitive and specific and can provide information on the likely cross-resistance profile. However, in many cases, markers for resistance are not available and they cannot be used. In summary, a validated molecular diagnostic can be used for resistance monitoring. However, they should ideally be used in conjunction with bioassays at regular time intervals in case new mechanisms evolve. We hope you have enjoyed learning about how we detect resistance in pest populations. Understanding when and where resistance arises is key to controlling some of the world's most damaging pests.